It's so nice to see so many friendly faces here, and this is arguably the easiest talk I'm ever going to give because I'm only going to talk for about 5 minutes. Then I'm going to hand it over to some of my colleagues at Boise State University, who it's been a really great privilege to work with. So today, what we're going to talk about is perfect. It's working great. Okay. Using the best available science to guide land management. So the city of Boise owns over 5,000 acres of open space reserves. We have 15 properties, and these 15 properties are connected by the Ridge River trail system, which goes over private land as well as federal land. The majority of our properties are in Sagebrush Steppe ecosystems, but we do have some property if you've been up to Stack Rock and Ponderosa Pine and Doug Fir. We also have a large greenbelt system, which is kind of outside of the purview of what I'm going to discuss today. But these properties are diverse, and these are actually owned by the city of Boise. I see many familiar faces here, many long-term conservationists, and so many of you were likely involved in the passing of the 2001 levy. It was a serial levy, meaning it was a 2-year levy where taxpayers in Boise opted to be taxed more to preserve open space to the tune of $10 million. And fortunately, many of those dollars were spent conserving very iconic properties that we have in Boise today, like Hall's Gulch Reserve, which is our very first open space reserve. But there was many other properties, as you can see on that map, that were conserved by the serial levy. Because we have such a forward-thinking, conservation-minded population here in Boise, there was another serial levy that was passed in 2015 with a 74% approval. And I included the ballot language on this slide because this is different ballot language than what was included in the 2001 levy. And you can see that the text is highlighted that improvement projects were included in the ballot language. So those are, it's not only for the acquisition of foothills property, but also for improving those properties. And that has lended itself to the creation of the Open Space and Clean Water Advisory Committee. The Open Space and Clean Water Advisory Committee is a committee of folks who are appointed by the mayor to serve on a committee to approve projects in that improvement project category. Anybody can apply for improvement project. If you Google Open Space and Clean Water Advisory Committee, you'll see that there's an application process there. And there's also conservation criteria associated with what your application is. So there's been a whole slew of different applications over the years, as you can imagine, but the conservation criteria is outlined, and that includes critical open space, clean water, native habitat and wildlife, trails or pathways, and community engagement. Along the bottom bar, as well as the larger slide, you can see what we've used the 2015 serial levy funds to buy properties. The ARS Stack Rock Boise is what I call the Stack Rock extension. For those of you familiar with that area, it's the west area of Stack Rock. Peace Valley Overlook, you might be familiar with that community based program where people living in that neighborhood actually raised funds and then the levy matched it, and that's how we purchased Peace Valley Overlook. Other properties, the Little Family, the 2020 property, that's how we have Antelope Bitterbrush Reserve, which is directly adjacent to Hidden Springs. Very beautiful habitat with lots of intact sagebrush as well as rare endemic plants. And some other properties that are there that we could talk more about. But along the bottom are also projects that have been funded by this improvement project stipulation. And you may wonder, why am I even talking about this? Well, this is how we've been able to fund science on open space properties. There's also grants, there's partnerships that we have with the city of Boise. But thanks to the levy and that additional stipulation of improvement projects, we've been able to fund science on open space properties. I talked a little bit about what was the conservation criteria, and this is obviously subjective. Your critical open space may not be my critical open space. But there is a committee out there that look at these applications, and this is how we acquire property, as well as fund projects that improve these areas. Something that's particularly important that I'm going to talk about a little bit later is the guidance documents for this conservation criteria. Where did we glean this conservation criteria from? Well, there's a handful of different documents, but one of them being the Open Space Reserves Management Plan, which you can also look at online if you'd like. It's a very outdated plan at this time. Some of you might have been involved in some of the community engagement associated with that plan. but it was a community-driven process where we went to the community and said, what do you want to see on your open space reserves? And specifically, I want to talk about this page. So goal 3.4 was doing research to obtain baseline data and track trends about the reserve's environmental resources and conditions and pilot innovative research programs. And so that's where we are today, is we need to utilize science to guide land management. And we're rare plant, you guys are plant people, we're all interested in plants, we're likely interested in science, but as rangeland management moves into the 21st century, managers like myself are increasingly expected to evaluate what types of management approaches are most effective for a given problem. And that's what science does. Science supports evidence-based decision-making. This is an infographic that I pulled out of a publication that came out in, I forgot the date, 21, thank you very much. And it talks a lot about adaptive monitoring, which is not what we're going to talk about today, but I want to take a step back and talk about adaptive management. And the term adaptive management was first coined in the field of natural resources in the 1970s. This is not a new term. And its roots can be traced to earlier concepts in ecology and environmental science. It emerged as a response to the realization that resource management often involves uncertainties, and that managers need to adopt flexible and interactive approaches to dealing with complex and dynamic ecosystems. While this concept itself was not originally tied to rangelands, rangeland management became one of the key areas where we talk about adaptive management. We talk about it all the time. And this is because rangelands are susceptible to very many unpredictable factors like climate variability, climate change, grazing, invasive species, soil degradation, urbanization, agricultural conversion. The list could go on and on and on. There's a significant amount of interest in adaptive monitoring to support adaptive management. And this infographic is from that journal. And like I said, we're not going to talk about adaptive monitoring, but I really like this infographic because it really shows the importance of monitoring as well as research to drive that decision-making that land managers like myself have to do. And then further, what are the major management issues that land managers like myself have to deal with? Once again, this can be somewhat subjective. My major management issues may not be your major management issues, but from my perspective as a land manager with the City of Boise, I am often overwhelmed with the idea of where to work. We have so many invasive species problems. We have so many different properties, and think about 5,000 acres. It's nothing in the breadth of a federal land manager who's dealing with hundreds of thousands of acres. But I am often struggling with where to prioritize where we put our limited budgets. Also, if you think back to that previous infographic, monitoring is a major component of adaptive management, and monitoring is also very challenging to do appropriately. Collecting data for the sake of collecting data is a colossal waste of time and effort. And from a land managerial perspective, I can tell you we've done that, and I'm trying really hard not to do that anymore. I want to collect data to drive management decisions. Getting that right is super challenging. And then on top of it, science is constantly changing, and the best available science is often different. So utilizing innovative techniques to combat some of the issues that we see on open space reserves is a major management priority. And something that's not on here that is perhaps as open space users and people who utilize trails, something that's also on my mind that we're not going to talk about today is the balance of recreation and conservation, which is a very challenging issue as the Treasure Valley has grown so much over the last 10 years. I really like this quote because I hope it gives you empathy for land managers like myself. This year is a really great example of a year where things did not go right for me. October was one of the hottest, driest months that I have ever seen on record. And we had contracts and plantings and all sorts of different treatments in place long before knowing that those weather conditions could occur. So I just show you this quote so that you can have empathy when you think about the stupid things that land managers are doing. It's really hard to pull it off correctly. We're going to end the talk today with a discussion about how it kind of went right with Elle. But I am super and incredibly honored to be working with Boise State University in many different capacities. We're able to fund and partner science with the levy as well as other grants and partnerships. And when you think about the major management issues that I'm confronted with, we have 3 different projects that I'm very, very proud of and very excited to be working on because they're innovative and also help us answer some of our management questions of where to work. Today, you're going to see 3 different presentations, one from Richie Holder, who's going to talk about soil carbon sequestration, another about goat head and invasive species with Richard. And then finally, the final presentation will be from Elle, who's going to talk about novel treatments and microbes in sagebrush plantings. Like I said, I am not the scientist here, I'm just the land manager, but I am super grateful to have this relationship with Boise State University, where young scientists can help inform land management decisions and meet local needs that we can directly apply to the ground. So everybody's gonna have to introduce themselves. So Richie is gonna introduce himself, Richard, as well as Elle, but thank you guys so much and thank you for your time and attention today. Thank you.
Just sharing. Do whatever you need to. I guess so we get this all squared away. I'm Richie Holder. I'm a graduate student here at BSU in the Marianne de Graaf's soil science lab. Thank you all for having me. I'm really looking forward to kind of sharing my research project with you today. And yeah, so let's jump on into it. So I know that we are a native plant society here, but today we're going to talk a little bit about the interaction between soil and plants. So before I get started, I would like to thank the members of my committee, Dr. Kelly Hopping, Dr. Derek Peterson, and my advisor, Dr. Marianne de Graaf. Their help has been truly immeasurable for me to even make it this far into the project. So I just want to say thank you to all of them first off. So the topic of my talk today is, there we go. It's going to be on soil carbon. So what is soil carbon? If you can look at this soil column here, you can see that there's areas of dark soil color in the column. So those areas have higher organic soil carbon content. That soil carbon can come from the top, the deadfall of the plant material at the top of the soil layer that goes down into the soil, and then alongside the roots that can release sugars, which are carbon, into the soil, and also dead roots that die and then can be captured into the soil as carbon. So just a quick refresher on the carbon cycle for all of those who do not know. This is basically, if we think about a bank account. So we have our savings account, and that can basically equal our carbon in the soil. So if we input more carbon into the system than what we spend, we're gaining money or we're gaining soil carbon. And the inverse happens. If we spend more money than we're saving, we're losing our savings or we're losing soil carbon. So carbon comes from the atmosphere into plants through photosynthesis. That carbon can then go into the soil through deadfall of, say, leaves or plant material onto the tops of soil, and then can enter the soil through that pathway. Or that carbon can go through the root system into the soil column. So when that carbon is located in the soil, microbes then come along and will then consume that carbon. Through that, they will then respire CO2 back out into the atmosphere. So it's this basically balance between inputs into the system, like our savings account, and then losing carbon out of the system, like our spending. So if we input more than we lose, we are gaining soil carbon. If we lose more out of the system than what we can save, we are then basically decreasing the amount of soil carbon we have. So why does soil carbon matter? There's tons and tons of soil carbon in the soil. There is 2,400 gigatons of carbon stored globally in the top 2 meters of soil across the world. That's 1 gigaton is equivalent to 134 million US homes, like their carbon produced. So it's a very large chunk of that carbon base that basically the earth has. So when that carbon is then released out of the soil, it can then exacerbate problems that we're seeing with climate change. So we can actually increase the amount of carbon in the Earth's atmosphere if that carbon that is currently captured in the soil then releases out of the atmosphere. This basically leads into the city's carbon neutral project by 2050. That is to basically find out how much carbon they're storing in their soils and then how much carbon they're producing through their actions. And we think that soil can be a very nice offset for this problem. So if you can see the small red dot located in the large kind of brownish circle, so that indicates how much annual emissions there are from the fossil fuel industry. And that is that 8.9 out of that 2,400. So it's a very small percentage of our total carbon that is stored in the soil. So we think that if we can increase the CO2 stocks found in the soil, that we can essentially offset emissions that we are currently creating. So to bring it locally, what is going on in our open space reserves? As we can see here, these are two different ecosystems. We have one intact sagebrush steppe ecosystem and the other being an invaded cheatgrass or Medusa head system. So we want to basically investigate how these two are different and then how we can inform land managers to better manage those areas for soil carbon. To do that, we would look at four different basically functional groups that we identified to basically help aid our study. So these functional groups were identified by the Ecosystem Services consulting firm. This is the vegetation map that the City of Boise uses for their land management. So inside of these inside of these functional groups, we identified four different major functional types. So that being the perennial grassland, which would be a mix of 3-on, squirrel tail, blue bunch grass, and then various forbs like arrowleaf balsam root, hoods, phlox, or hoary tansy aster. We have upland shrub category, which was our sagebrush and our antelope bitterbrush, with also some annuals and perennials kind of located in the understory of those plants. Then we have our annual tall category, which is predominantly comprised of cereal rye, which was a formerly planted species by the city, but is now kind of escaped and basically is of management concern. And then our last category is annual short. So this is where we have our more common invasives like Medusa head, cheatgrass, and other kind of noxious weed species. So I do want to say as just kind of a caveat that almost all of the sites that we sampled out in the foothills have some level of cheatgrass and Medusa head invasion. It just kind of depended on the basically severity of that invasion at that site. So why were these functional types basically identified and why do we use them as proxies for soil carbon? So if you can look on the far left here, you will see that we have a sagebrush. This has a very large above ground biomass and also a massive taproot that goes very deep into the soil column. So that also has a lot of below ground biomass stored there. And inversely, over on the far right-hand side, we have an invasive plant like cheatgrass that has a very, very relatively small above ground biomass with a very tiny root structure. So these are kind of the two opposites of the spectrum. On one side, we have our upland shrubs. On the other, we have our cheatgrass, and our other plant species kind of fall, our functional types fall somewhere kind of along that continuum. So as we look out onto the Boise foothills, we can see a very complex landscape, both in the macro and the micro. In the micro, we can see that there's many different types of species located. We have bare ground, we have rocks, moss, and different kinds of plants in terms of whether they're a perennial or an annual. We also have that same level of complexity across the whole landscape scale. So that being our different aspects, soil types, and the elevation gradient. So kind of all these factors together are what we use to basically come up with our sampling regime. So we are going to talk about that now. These are the 7 sites that we chose to investigate. We have 3 sites that we categorize as kind of a high elevation reserve, that being Noble Reserve, Hawkins Range Reserve, and Stack Rock Reserve, with 4 sites being our lower elevation. So Military Reserve, Camel's Back / Hulls Gulch, Hillside to Hollow, and Polecat Gulch. So we are going to take just a closer look at Polecat Gulch Reserve quick here. So we can see these were the different sampling points that we identified to basically conduct our project. So how we came up with those points is we took the vegetation layer here from the City of Boise's basically vegetation map. So this has our 4 different major plant functional types, that being our annual shorts, annual talls, perennials, and shrubs. So then we also overlaid that data with our different soil types that we chose to investigate and the aspect. So kind of all these points that we found were different combinations of those three factors rolled into one. So I'm gonna see. So we chose to, let's see if I can get this. Oh, there we go. So when we rolled into one of these sites, we would establish a 10 by 10 meter transect. We'd then put a center point down and we would take a soil core underneath one of those plants that we identified. So if it was a shrub species, we would take a soil sample from underneath one of those shrubs. That soil core was then split up into 3 different sections, that being the 0 to 10, 10 to 20, and 20 to 30. So that allows us basically to see different carbon levels as we go deeper into the soil. So we expect there to be more carbon in the upper layer of soil than there would be in the lower layer of soil. And that's kind of identified by the different hues of color that we see on the screen here. So the physical nature of this project cannot be understated. I am forever indebted into the great folks that worked on me with this project. We hiked in total about 173 miles and we collected about 1,130 pounds of soil throughout the project. So truly the blood, sweat, and tears from these good people. One of them is here, Arden. Just want to thank you for your work on this project. It was truly could not have been accomplished without you. So this is our basically photos of our sampling regime here. So we can see on the far left, we have very large species. This is from one of our shrub sites, and it's also on northwest aspect. So those are larger plant species due to the higher moisture content. In comparison to the middle photo here, we can see this is one of our annual shorts or more our invasive sites. And from a southwest aspect, so due to the lack of moisture, basically very small plants, so we expect there to be difference in soil carbon between these two. Then on the far right, you can see a picture of a soil core that I took as my entire sampling crew is working very diligently. I paused to take this nice photo, but to put it up there, it kind of shows the difference in soil color as we go down the column. So we can see on the far right, it's a little bit darker hue. That means that more kind of soil carbon content. Then as you go deeper, there's kind of that less coloration in it. So to process all that soil, once again, took a small army of people that I'm forever indebted to. The process for basically processing the soil was to dry the soil for 48 hours. We would then take the soil sample and put through a 2-millimeter sieve. We would then take a tweezer and remove all the roots and the rocks out of that soil sample. So we were just left with bare soil. That was then basically subsampled, ground into a powder, and then analyzed for soil carbon. So this is what our soil samples would look like. We can see that there's a mix of different colorations of the soil, different amounts of roots in those samples, and also different amounts of rocks in those samples. We can also see a difference here between our higher elevation and low elevation soils. So the one on the left-hand side is from Noble Reserve, and then the one on the right-hand side is from Hillside to Hollow. So the one on the right is from one of our lower elevation sites, and the one on the left from one of our higher elevation sites. So, and with that, our major findings from this was that 1, shrubs store more soil carbon than all of our other plant functional types. This was across the board, combining all of our plots or all of our samples into one great bin. We found that shrubs were by and large the most important takeaway from this, that they store more soil carbon consistently compared to those other functional types. We also found a very neat relationship between soil carbon and the distance from a trail. So the further off a recreational trail that our plot was sampled, we found more soil carbon kind of underneath those spots, and that kind of just indicates a little disturbance due to basically recreational activities. So we also found that there was a different impact of vegetation across basically the different reserves that we investigated. So we found that there was 4 reserves that had shrubs being a very significant impactor of soil carbon. And across those reserves, each of them were our 3 low elevation reserves, that being Camel's Back, Military Reserve, Polecat Reserve, and Hawkins Range Reserve, with Hawkins Range Reserve storing the most carbon out of all those. So my big takeaway from this entire talk here is that shrubs are key to soil carbon when it comes to our system. So with this data, kind of an endpoint for this project is we are going to be making basically a map of soil carbon. So this is a similar map that we're going to be creating. This is from the Rounds Creek Experimental Watershed. So this shows basically how much soil carbon and where it's being located. So the endpoint is that we're going to create this map for the city, and the city is then going to be able to use that as a resource to kind of highlight areas of basically need of management or prioritize their management practices. And yes, thank you all for paying attention here. Appreciate it.
So I have a shameless plug, Russ and Janet Burchard. Thank you for helping me with that. Yeah, they have the most beautiful native plant garden I've ever laid eyes on. They're also some of the kindest people that I know. Russ is an incredible woodworker and has made these really beautiful bookmarks out of sagebrush and bitter brush. Now that we know that shrubs are so important for storage of soil carbon, they can also help you mark your books. So I was gonna pass these around. He's been kind enough to leave a box of them in the back. Thank you. Yeah. And if you have questions about his work, you're more than welcome. I have one. I value it. I treasure it. It's highly recommended. Thank you so much, Russ and Janet, for everything.
That leaf hackberry, and this happens to have some spalting in it. Gorgeous, my favorite tree shrub out there. We'll pass some of these down. And these are all from dead. It's a trick. You gotta get them fairly soon, otherwise they start cheating.
Oh, I'm sorry.
Yeah.
Whatever you're gonna tell
Yeah.
us.
Zane, go to the first one. I guess
I've had one speculation.
I can just use the.
I'm a mountain biker and I don't like the idea of my
Right.
trails causing more carbon.
Yeah.
And is that because we're tracking
You won't
more
go.
invasive
And is
species
this gonna, this is already. up.
along the 20, 30 feet away from the trail?
Oh, cool.
Yeah.
Is that the reason that there's less shrubbery there? So if
Yes,
there's
absolutely.
something like that, there might be a cause and effect. going on.
Yeah,
I don't have to pick it
so
up anyway.
unfortunately, this project really wasn't designed around that distance from trail kind of components. So we really can't necessarily answer the question of. Yes, absolutely. Thank you. So yeah, the question was all about kind of the distance from trail. So unfortunately, this project kind of found that as a takeaway more so than a question that we kind of originally sought out to answer. So I think it's a great hypothesis as to there being kind of more invasive annuals along the trail system due to, folks hiking, things of that nature, kind of carrying seeds. But we unfortunately don't really have the data to back that up. I mean, that can be a future project that, the city can investigate to see kind of how that disturbance impacts the soil regime and things like that. Yeah, and any other questions that anyone has?
Did you account for the functional groups with that finding?
Yeah.
Like despite the fact that it's.
Yep.
Grassman.
Yeah.
And then did you double check when you did your course, like what functional group you were in?
Yep. Yeah, absolutely. Yeah. Every single core is just kind of labeled for what vegetation and what vegetation group it came from. And then the samples that we would take would be directly underneath, the corresponding kind of vegetation functional type. Yeah.
Was there a particular substrate that seemed to hold more carbon than another?
Yeah. In terms of the soil type
Yeah,
or
like in decomposed granite versus.
Yeah.
What happened?
So our soil types were kind of basically the classification split that we did was off of the slope and off of basically the carbon content due to clay.
Oh, okay.
So that was kind of our justification. So unfortunately, I really don't know what the kind of parent material would be for those, but we did find that the Haplixeral soil type that has the highest basically clay contents
Oh, okay.
stored the most soil carbon out of the
So that's
groups.
kind of like the hillside to hollow area, super clay compact.
Yes.
Okay.
Yep. Yeah.
You said at your plot, I thought you said you collected the sample underneath one of your shrubs.
Yeah.
Well, how do you even that over the whole area at that point if you've biased it to under a shrub? Not
Yeah.
all the area is under a shrub.
Yeah.
Actually, only a small portion of it is under a shrub, so
Yeah.
only a portion of it has, that plot has that level of soil,
Yeah.
carbon.
Yeah, and so we do expect there to be basically a higher carbon content, like right next to that root system. So basically, we would then, in our carbon model that we're going to be making, basically account for the vegetation map that we currently have and use that as an input into the carbon model. So that's a way that we can basically account for the difference in vegetation community based off of this vegetation layer that we have here, as a way basically to input and almost justify in a certain way why we have those differences across carbon in terms of the vegetation community.
So let's say the sagebrush and bitterbrush have burned. Do you know, have any idea how long that carbon, the deep root system stays in there?
Yeah. Unfortunately, I actually do not know in terms of like the fire history and what that has to do with the root system of those plants. Yeah, unfortunately, I study the soil carbon. Yes.
I'm a little shocked that the native grass orb component was just a little bit better than cheatgrass.
Yeah.
Do you even have healthy native forage grass?
Yeah.
out there?
No, that was in our original hypothesis, that was kind of our takeaway, is that we thought that there would be a higher amount of carbon due to, or stored in the perennial grasslands and upland shrub in comparison to our two annual mixes of those vegetation groups. So yeah, that was definitely a shocking result that kind of we came away with. And I really do agree that it probably is due to that the lack of actually intact bunch grass or kind of forb systems. I mean, a lot of these areas had cheatgrass, predominantly in the understory. Really, no matter where you are in the Boise foothills, that was kind of a factor that we were constantly dealing with, was that invasive grass elements. Yeah.
In geologic terms, cheatgrass invasion is relatively recent.
Yes.
So then I'm wondering how many centuries, or whatever, are we talking about in terms of buildup of carbon in these areas?
Yeah, no, the time scale is a really interesting component. In terms of the soil carbon, it's a very, very long time scale in terms of almost the thousands of years, in comparison to a more kind of rapid time difference of that being the above ground biomass. So kind of how these two carbon pools move is drastically different in between the above ground, below ground kind of systems. So it's definitely something that needs to be studied more of a long-term time scale, I would absolutely say. Kind of the impacts of these things over time as we go through, say, 50 years, 100 years, something of that nature. Yes.
So I did a little experiment out in Oregon a while back, and I noticed that sagebrush, when we break it, you get a little like a puff, almost like smoke, right? And so these are all aerated shrub steps, right? So basically you need a little piece of carbon to fill that void to retain water. Right, so my question being, would there be more carbon in places where there was dead, decaying sagebrush or so on, or even water level?
Yeah, no, absolutely. Yeah, definitely more carbon is associated with basically that sagebrush biomass. And a lot of that is due to kind of the chemical composition of that shrub. So it actually breaks down a lot slower in comparison to, say, like a grass stem, where that will basically decompose much, much faster than, say, a shrub, root or a branch, have you. And then water is also a fantastic predictor of soil carbon storage. Basically, the more productivity an area has, the higher carbon that's going to be found in the soil.
Got two more.
Yeah, I don't like my face. Go away? Yeah, cool. There we go. Hi, my name is Richard Rachman. I was supposed to talk about goat head, which please come talk to me about goat head. I love talking about it in our goat head hotspot map. But so yeah, come talk to me. I have tons of presentations. I could whatever talk your head off. But today I'm going to be talking about some kind of new results and some preliminary data that we're currently working on with some of our drone work. So quantifying spatial clustering of native shrubs after wildfire. Again, my name is Richard Rachman. I got my master's at Cal State Northridge. I was also the volunteer coordinator for the Western Monarch Count with Xerces Society. I do a lot of cool stuff. Cool. Here's my Instagram. I'm in Trevor Coughlin's lab at BSU. as well. Okay, so like, might need this a little bit. Can I stand over here? No. Why does clustering matter, right? So when you look at an arid plant community, often in the desert, you'll either notice that plants are very closely together or often far apart, depending on how much water is around, what elevation you're at. So if you were to go somewhere like in the Creosote Desert, in the Sonoran Desert or whatever, you'll notice the shrubs are not very close, right? They seem to be competing. But if you go up in elevation just a little bit, you might notice some shrubs using other shrubs to kind of like help each other out, right? So we have these things that are going on that we can ascertain about plant populations based on how their spatial arrangement is. So here's a figure that I really like from a 2022 paper. If you look at a bunch of shrubs and they're kind of like randomly distributed, or if they're aggregated, or perhaps they have kind of like really normal, regular dispersal and like spacing, right? And that could tell us something about the ecology of those different patterns. Or perhaps it's kind of random and independent of other species. Or perhaps certain species or the same species are attracted to each other. Maybe there's some sort of fungal mutualism going on. Or maybe there's like a plant that has allelopathic compounds that's like shooting off and like kind of shooing away other plants from it. It's repulsing other plants. So you have facilitation, right, where plants could be helping each other. Then you have competition, where plants could be repelling or kind of competing for resources or trying to get other plants in somewhat antagonistic relationships. So there's a Mariposa lily I just wanted to highlight that like you'll often find under sagebrush these really pretty forbs. So it's an example of kind of a host-plant relationship. There's a cool picture I found of a bison with some lovely earring on of a sagebrush. But so how does like disturbance come into play here? We were talking about wildfire, right? So like often in arid ecosystems, wildfire only penetrates just a few centimeters of the soil, but it does have really big effects for above soil biomass, right? It can totally knock out a ton of the available biomass in an area, cause tons of erosion, and that can affect the soil down below. But wildfire is a huge disturbance into an ecosystem. You also have humans that are, not helping the situation, spreading invasive species, or perhaps just like with our recreation or trail use. And then you have animals, right? Historically, we had huge populations of ground squirrels everywhere. They were digging holes and rodents that were doing lots of things, and we pretty much killed most of them off. So you're having changes in disturbance that would positively or negatively be affecting plant populations, and those disturbances are different now. In those invasive species that humans have been bringing into places like Idaho too have had an effect on not only how plants are distributed on the landscape, but also how plants can recover in the landscape as well. If you have more invasive species, you're going to have more wildfire. Cheatgrass and Medusa head promote wildfire, and these are going to create fire return intervals, which historically we didn't see before. So we have areas that get burnt. What do we do, right? Like we had the Valley Fire, and they're like going up there and doing like tens of thousands of sagebrush plantings. Like, and a lot of us are kind of like, most of those are probably going to die, right? It's not very encouraging, but one thing that's coming out of the arid ecosystem restoration ecology literature is this idea of islands, right? Where we do restoration in large patches, right? And they kind of be this perfect size in the perfect location. You have to look at this figure from, it's a US Forest Service paper from 2017, really good paper. But this has this kind of infographic. You get all these environmental variables, right? What's the soil like in the area? What's the topography? What's the vegetation that's already existing there? Get like a management strategy for like where you're going to do your restoration. Are we going to use herbicide for these invasive species in just this particular area? What herbicides you're going to use? And then you create your islands, right? And they kind of have to be this kind of perfect size in order to either do one of two things: spread from the center and spread outward, or spread in such a way with certain types of dispersal of plants that they spread and make more new islands. So it really depends what your long-term goals are and what palette of plants that you have to work with. But it's a really interesting idea when it's super hard to get water to these restoration sites in order to do restoration, right? Yeah. But I've also seen literature like supporting the use of dead shrubs in order to create these islands, in which you can use like dead plants in order to put baby plants around them in order to create your island, which is also a really interesting idea. But so space matters. The arrangement and pattern of these plants matters. So are some questions that have been informing our research. How do wildfires alter spatial patterns in native shrubs dramatically? What environmental variables are important for altering spatial post-wildfire patterns? And then how can we use information of spatial patterns to inform restoration, right? Science has to mean something. It has to have an end goal. And that's kind of what we would like to see as well. So we have our sites. As you see, the happy rainbow is like kind of where Boise is, not really, but whatever. So this is a map of our 14 sites that we used drones and flew over and collected our data. So on all the different colors of our sites are along an elevational time since fire gradient. So the lighter or more recent fires and the darker ones are more like older fires. So it's about from an interval of like, we have a fire from like 7 years ago to about 26 years ago. So, which is shorter than the historic fire return interval for Sagebrush Steppe, which can be between like 50 to 150 years. And then numbers vary depending on who you ask about that. And then we also have elevational gradients, right? Because in Sagebrush Steppe, the higher elevation you go, the more snowpack you have, the more water you have. And so you would think that that would affect how shrubs are kind of aggregating as well, and how they're going to recover post-wildfire. So that went into our modeling as well. And then so the dominant plants at our sites were mostly, the rock stars, sagebrush, rubber rabbit brush, antelope bitterbrush, and these 3 shrubs are the ones that I'm going to show you results from today. Like I said, this analysis is ongoing, but we have some initial results we're excited about for these 3 shrubs. There's my iNaturalist observations for them. Go download iNaturalist if you don't have it. Some other natives, we have wild rye, green rabbit brush, low sagebrush. There's a ton of others. We only measured things that were typically taller than 25 centimeters, and this is due to the ability of the artificial intelligence to identify the plants, which I'll talk about in a sec. And then we also measured some like patches of non-natives, and we did this in collaboration with Megan Gatau's lab, they're looking at drones and mapping invasive species. So we measured things like cheatgrass, tumbleweed, intermediate wheatgrass, some other stuff as well. So the drones, right? So we had multiple drones for all these 14 sites. We had multispectral, which is usually 5 color bands, but some of our drones were 10 bands. And then we had drones that were cheaper, not as expensive, that were red, green, blue. And that's like mostly what you're going to be. If you're doing drone work, that's like most of the drones you're going to buy. From that red, green, blue, we could also, we took thousands of pictures of our sites. We assembled the pictures in such a way that we could create 3D images of each one of the plants. And so that gave us vegetation heights as well. So you didn't actually have to send a field tech out to measure the plant. We could get like really accurate measurements ourselves. I did go out there though. And so you can see one of our GPS devices out there, our TopCon. And with the RTK unit, I'd walk around to a shrub, record its location, and then go to another shrub, record its location. And we did this hundreds of times for the different sites. And these were training points for the artificial intelligence that we use later, the machine learning algorithm to classify the imagery from those centroids. So the drone has incredible resolution, right? For red, green, blue, it's 1 or 2 centimeters of resolution. So your satellite imagery is typically, if you've got a lot of money, it's going to be about 1 meter. If you're just using free stuff, it's going to be like 3 to 10 meters, depending on what satellite products you're using. So we can get really high resolution pictures. And then with our GPS units, they're about 1 to 2 centimeter accuracy, which is really good. Yeah, and so then we use those training points to classify the imagery as well. If you want to know more about that, you can talk after. Why isn't that going next? Technician. Oh, there we go. Okay. I did it. So this is kind of what that classification looks like. So if you look at those teeny little pictures over there, so that's like a zoomed in picture. This is Sinker from the Sinker fire, and that's out by Melba. That kind of, if you look at the image, that kind of like weird river looking thing that splits the image is the where the firefighters got their equipment out there. So that's the fire line. So mapping these, like what's the burned area and what's the unburned area was kind of easy. All of our plots had these kind of burned and unburned areas. So we could do like a comparison between the plots. And so what we did was from the drone imagery, we could then classify what the shrub was, what were the dimensions of the shrub, how tall it was, and then draw canopies over them. Now, they're not always accurate, but they're pretty good. Especially like I'm looking at this screenshot that I just haphazard, like I just zoomed into the first place. I was like, okay, good, I can share this at the presentation. It works, it's pretty cool. So here's our preliminary results. So this is showing something called Ripley's K factor, and Ripley's K factor is like the intensity of clustering or dispersion. So basically the only plant of the three that we looked at sagebrush, we looked at rubber rabbit brush, and we looked at Persea tridentata, the antelope bitter brush. Rubber rabbit brush was the only one that has statistically significant result. And it was in the unburned areas that it was really dramatically dispersed. I'm sorry, in the burned areas. In the burned areas, it was significantly dispersed. So plants of rubber rabbit brush were kind of far apart from each other. And that was the one result that we have so far using this Ripley's K analysis. We didn't find anything significant in terms of elevation interval for any of the species. So time since fire or like at what elevation the site was. And neither of them had that dramatic of a difference in burned and unburned for sagebrush and for antelope bitter brush. We suspect this is going to change with more analysis. We're going to do something called a replicated point pattern analysis, where sometimes in experimental designs, you'll have replicates, and they could be like part of a site or something, or a treatment, statistical treatment they're using. For our replicated point pattern analysis, in which we look at distances between plants, every single one of our plants is going to be a replicate. So that'll give us a sampling size of 100,000 to 200,000 plants for our analysis. Those are all plants that like you didn't have to have a technician go measure. Like we have all that data from drones. That's kind of another cool reason for drones. Artemisia tridentata and Persea tridentata. I should have probably written out those scientific names, but like my work as a as a botany tech just kind of takes over sometimes. And I use veg codes. Yeah, so those are veg codes just so we can write faster. Why rubber rabbit brush? Like what's so important about it? Why are we getting this result about this? We don't know. And it's a really quirky result, but also rubber rabbit brush is disturbance adapted. I see it in abandoned lots all around Boise. I see it persist after wildfires. It'll actually like sometimes re-sprout. It's not definitely a fire follower per se. Like a lot of California native plants will persist after wildfire, and they're promoted by wildfire. I would not say that about rubber rabbit brush, but you know, it will come back. And so perhaps we could leverage this, right? If we're focusing plant restoration on disturbance adapted plants, things like milkweed, things like rubber rabbit brush, grasses like Sandberg's bluegrass, things that we know pop up after wildfires. What if we focus on those for restoration? And then the sagebrush will come in by itself later. Or maybe once this ecosystem's established better with more native plants, we could then reintroduce sagebrush after we've gotten those like early successional plants in. It's also wind dispersed, so that could be a reason why we're seeing like such dispersion in the burned areas. And it's kind of like random dispersion, so that could dispersal kind of explains for that. But also I want to make the argument too that this is also a nurse plant. Like sagebrush could be growing underneath a rubber rabbit brush. So they're really important plants that are kind of like slept on in terms of native plant restoration. And I'm going to make another argument why I love rubber rabbit brush. They're huge attractors for native bees. So I did a survey with a bee ecologist. Her name's Crystal Hickman, or BSIP on Instagram. Her photos have been featured on like Nat Geo and stuff like that. We went out to the Sawtooths. We just did a 4-hour survey, and we found over 20 species of native bee in that 4-hour survey. And we didn't kill any of them. Her whole shtick is just live photography. And most of them were on rubber rabbit brush. And if you've been around a rubber rabbit brush and you just hang out with it and it's like late summer, early fall, this won't surprise you. They attract so many pollinators. And if more bees are becoming listed as the days go on, like we have a bunch of bumblebees that are listed, this problem for native bees is getting worse and worse. So this is another reason why we could be looking at disturbance adapted pollinator plants for our restoration sites, and how they're arranged on the landscape. Maybe in these islands, it's kind of undetermined. We're not really sure about the islands yet. But I want to thank a whole bunch of people that I'm not going to name. Maybe you see your name up there or something. But I'm financed by the NASA Finest program and the National Science Foundation, and I've received a scholarship for the Idaho Native Plant Society, so thank you so much. There's my advisor and some of my lab mates that have helped out with the research. I'm going to make a call right now. I'm looking for a job. I'm looking to go on to greener pastures. There's my LinkedIn QR code. I do a lot of stuff. I mostly do geography work, so GIS and remote sensing, but I'm also trained as a botanist and like focusing on issues that affect LGBT people and DEI issues in the workplace. So there's my email address, my LinkedIn, and thank you so much.
A pleasure to work with and very responsive. I highly recommend hiring Richard. With that said, we're pressed for time, so I'm gonna have Elle come up and talk. Thank you so much, Richard. I talked about how land management and adaptive management is very challenging. Because of feasibility. Elle is going to talk about microbes today and a little bit about her research, but we had a very happy accident associated with managing the polecat fire, which only burned 32 acres. It was very small in the spectrum of massive fires that occurred. But I grew out, we did sagebrush seed collection from Polecat Reserve in 2023. Those seedlings were grown out by Lucky Peak Nursery, and Elle was able to do her research on those seedlings, which she'll talk about more, and then we planted those seedlings into the polecat fire. Did I plan any of that to happen? Absolutely not, but it was a very happy accident that occurred in land management. And so I'm very excited about what Elle's results will yield. It's very seldom that one collects seed from a landscape that burns, has seedlings available for the restoration of said landscape, and is able not only to put those plants on that landscape, but to do research as well. So most of the time, we don't get it right, but sometimes we do. So thank you so much, Elle.
Thank you, Martha. Okay, hi everyone. I'm Elle. I'm a PhD candidate at Boise State, and I work in Dr. Leonora Bittleston's lab. I do research looking at chemical and microbial ecology of native shrubs. I will table the chemical ecology for now because this dissertation work is solely really looking at microbial ecology. So to get started, we're going to be talking about some novel methods for using microbes in sagebrush outplantings for restoration. So some background information. I will not go into too much detail, but microorganisms, generally microscopic organisms, it can be single-celled, multicellular organisms, think like bacteria, fungi, viruses, archaea, protists, like all these things are,
Sorry.
all good. I'll just keep talking. So really fun fact about microorganisms is that they are actually the most abundant organism on Earth. And they are incredibly important for ecosystem processes. So luckily for me, Richie started this talking about the carbon cycle, and he had those cute little animations of Pac-Man, which were the microbes helping with the carbon cycle. So they play crucial roles in nutrient cycling, say like decomposition, and overall are really important for ecosystem health. So microbe-host relationships. So microbes can form anywhere from antagonistic to beneficial relationships with host organisms so think I'm sure a lot of us already know about, say, our gut microbiome, and that's how important that is for like our health as humans. I'm not going to talk about all of that stuff, but what I am going to focus on is plant-associated microbiomes. And so roots, stems, flowers, and especially leaves, which is what I'm going to talk about, are all plant-associated microbiomes and have implications for plant health. So examples of this would be plant growth promoting bacteria, which is one we're going to talk about today, which can enhance nutrient uptake, produce growth hormones, or suppress pathogens and diseases for certain plants. One that I'm sure a lot of us are familiar with as plant people is the symbiotic relationship of mycorrhizal fungi and how they can form these relationships with plants to improve nutrient and water absorption. So leaf associated microbes. So what you might not know is that leaves can actually harbor these diverse microbial communities. The picture right there is actually sagebrush leaf microbes. It was a plate cultured from a couple summers ago. But microbes can vary depending on plant and environmental factors. So say they have important things for, like, they can change versus on environmental factors. So humidity, temperature, UV radiation, these different microbiomes can also change between different plant species or even within the same species, but just between individuals. They can perform important functions for host plant health, and so defenses against pathogens and herbivores, nutrient acquisition, or drought tolerance. So, microbial inoculation in agriculture. So this is something that dates back to the 19th century, and it is this, where beneficial microbes are applied to soils or plants with the goal of improving plant productivity and crop health. So thinking about increasing plant growth and yields in an agricultural system. So biofertilizers are used to enhance plant growth and nutrient uptake. This one, EM1, is actually one that is available on the market right now, that has just a certain type of lactobacillus that is used for plant growth. Biopesticides are also used in the agricultural market to suppress plant diseases and pests that can cause problems for crops. So the benefits of using these microbial inoculums can be reduced reliance on synthetic fertilizers and pesticides and improve plant growth and yield, which is becoming an increasing issue as we have more people on our planet and we need more food. Okay. So, plant people. Big sagebrush, that's what I'm going to talk about today. And I'm not going to go into a lot of detail because I'm sure we all know it's a foundational species in the Western United States. It's declined by 55% of its historic range. The contributing factors to this could be invasive species encroachment, increasing wildfire intensity, and land use management. And this all has other implications on things in the ecosystem, like all the threatened and endangered species that also inhabit this ecosystem. So, sagebrush-associated microbes, they harbor these microbial communities on the leaves, and so the lab that I work in, we look a lot at these microbes, and we do research on these microbial communities and maybe how they temporally change over the span of a year. So one of the PhD students in our lab, Jacob Heil, he's done some really cool work looking at how these microbial communities change throughout a year. He also did some work characterizing the sagebrush microbiome, like these internal and external microbes, and ultimately had these isolates of 155 fungal and 27 bacterial cultures just from local sagebrush leaves. So not his work, but also some more sagebrush leaf microbe pictures. This was just some preliminary work that I was just doing to collect some microbes that were from sagebrush. And these plates were just like set out for 12 hours. And we have so much microbial growth that's just from these sagebrush leaves. Another graduate student, PhD in our lab, he does stuff with metagenomics of sagebrush leaf microbes and thinking about how like individual microbes might perform certain roles for sagebrush health. Okay. So going back to Jacob's work, he had those bacterial isolates that he identified from sagebrush. One of them was Bacillus amyloliquefaciens, which I'm going to call it B-Amy from now on. But this is this endophytic plant growth promoter in other systems. We have not actually identified whether it does the same thing in the sagebrush system, but that's what I'm going to talk about with you all today. But this versatile microbe, which it can be pictured on the right, is this rod-shaped bacteria that can produce antibiotics, some enzymes that like break down fungal pathogen cell walls. It can produce these plant growth hormones, so like auxin, and also like can make or phosphorus available for plants through like phosphate solubilization. So it's already, this bacteria is used in agriculture. It's already widely used in an agricultural setting. But it's also happens to be native to the sagebrush leaf microbiome, which is very cool for my experiment. So as we've already talked about, the replanting success for sagebrush is often low. It can be anywhere from like 15 to 20%. 20% is good from Martha's words. So the thought behind this experiment is to really utilize this one bacteria to see if by introducing this microbial species to seedlings as they're growing before we outplant them could improve the survivability and restoration outcomes that we see when we go and plant sagebrush. So the experimental design, which Martha's already touched a little bit on, is these seedlings. They were seeds collected from either Polecat Gulch or Hillside to Hollow, which I did not help with that. She did an amazing job. She had all these seedlings growing at Lucky Peak Nursery, so also shout out to them for helping me do this work. But these seedlings were being grown, and so I utilized this opportunity with Martha's help to randomize and inoculate with this bee Amy. And then once they were outplanted, the plan is to then track survival. So also a lot of the stuff is going to be preliminary because we just did this planting in October and the holidays. So I didn't go out and check in December. But then we're going to track survival over the year to see across these treatment groups. So treatment groups, I'm going to do some breakdown on like a little bit of math so that you guys actually know how many seedlings were actually happening. So, there were these trays that you can kind of see in this picture right here. So this is like the setup at Lucky Peak Nursery. The little white thing is a tray where all these seedlings are being grown. And so each tray can carry 112 seedlings. That is if 100% survival of all these seedlings. Of course, that doesn't happen. That would be incredibly unrealistic. But these seedling trays were randomized into treatment groups, control groups, or negative control groups. And for each location, there was one negative control that had nothing done to it, except had pictures taken as they were growing. Then 3 treatment trays, and then 3 control trays. These treatment trays were treated with bee Amy. They were sprayed with an inoculum that I prepared, and they had an active culture of bee Amy in them. The control were sprayed with sterile water. There was nothing in it. It was sterile. I checked, which is also why in this photo, you can see that this is me testing stuff and spraying. So the T is the one that was with the treatment. And so those little white dots are bee Amy when it's grown up. And then the C is just a very blank plate, which funny enough, kind of looks like it has stuff on it in that, but I swear it doesn't. I swear it doesn't. It does have a flash on the camera. I think it's like a little glare. So anyway, so all of these were sprayed, and inoculations started in April when the seedlings were very young and just had new fresh growth on them, but hadn't actually like grown above the tray lid, so just about 4 centimeters or so. And then this was done on a bi-weekly basis until October, which is right before they were outplanted. So the planting sites, like we've talked about, was Polecat Gulch or Hillside to Hollow. They were planted back at wherever their seed location origin was from. So for Hillside to Hollow, we had a total of 621 seedlings planted. For Polecat Gulch, we had a total of 569 seedlings planted. I cannot go on without saying how much help that took, and I'm so grateful for all the volunteers who came out and helped me with that. And Richard also was brilliant and organized a volunteer group for the Hillside to Hollow, which was amazing. And then also Megan Cateau's class, the UAS drone flying class, also came out and helped me with the Polecat Gulch site. I could not have done it without them. It was still like 14-hour days of planting, and it was very, very hard. So I couldn't have done it without them. So also talking about what Martha was talking about with the Polecat Gulch is that area recently burned in the summer. The seeds were collected before the burn. Burn happened, and then this is also kind of a unique opportunity where we can do some post-burn restoration and see about the success of post-burn restoration. Yeah.
Okay, what time or when, and then how deep were the roots?
Ooh, great question. When we planted?
Yeah.
We planted October 9th and 15th. Pretty sure that's with 9th for Hillside to Hollow and 15th for Polecat Gulch. And roots?
There were 5.5 CI. The cubic inches they were on those styrofoam blocks the size of 5.5 cubic inches, so not a lot. I mean,
6, 8 inches deep?
yeah, I would say approximately 6 inches.
And it was highly variable. Some of them
Water?
definitely
Water gets the final.
no, which is which yes, Martha and I had discussed. It's the idea behind this project is also, if it is successful, it has to be realistic for the people who are going to be doing these plantings. And so watering everything in, especially at numbers like 600 to 500 seedlings, is just not really realistic. So the goal behind it was, I asked Martha, how exactly would you do this? And like no special treatment, what would you do just from a land manager's perspective? How would you treat these plants and plant them? And so that was what we did.
Why October was so rough for me.
Yes, we did have multiple conversations where it was very much just like, this is not the ideal, especially for Hillside to Hollow, maybe not the ideal planting conditions, or yes, the soil was also not great at like Hillside to Hollow. So data collected so far. Shout out to Chadwick. Chadwick helped with some of this data. So we have initial canopy area and final canopy areas, all calculated by Chadwick. He did a fantastic job. And so I'm going to show some preliminary results on the next slides, thanks to him. We also did some external and internal leaf cultures. If you want to see any really cool pictures of microbes, I've got a lot. And that was done with help with Ade. And then we also have GPS coordinates for all the outplanted plants and drone data across the Polecat Gulch site. Not for Hillside to Hollow, but for Polecat Gulch. And so special thanks to Megan and her class. So preliminary results, these are very preliminary, but plants treated with B-Amy did show slightly higher canopy area. It was statistically significant, but I also would like to dig more into like if I'm actually looking at the results correctly. And so right now, the treatment kind of works. Future aims are going to be to continue collecting drone data for Polecat Gulch, which also means like ground truthing by tracking survival and growth over the next year. I'm going to try and do it on a bi-monthly basis. And then also connecting plant survival to slope aspect, trail proximity, especially for Hillside to Hollow, since that is also an off-leash area for dogs, which also like increases disturbance around trails. And maybe over a burn severity gradient. Something else I realized that I didn't add to the slide, but we were kind of talking about with burn skeletons. So because the Polecat Gulch burned, there's also lots of sagebrush that were living there that are now burned. And so it would be, I did intentionally plant at a varying distance to certain sagebrush skeletons at that site, which also means that I would like to also incorporate some proximity to a burned skeleton, because maybe that carbon storage is helpful for restoration outplantings, especially those post-burn sites. I feel like there was something else I was going to say, because Richard was talking about the clustering. And so also the way that we did plant, they are technically in clusters. And so hopefully that also might increase success. And at the Hillside to Hollow site, we were intentionally trying to bridge 2 islands of sagebrush. So we'll see how that goes. Anyway, this is my acknowledgements to all of the amazing people who helped me do this work. So special thanks to Martha also for taking a chance with me to do this.
Thank you so much, Boise State University, and thank you guys, Richie, Richard, Elle, for doing all this really important science that helps guideline management. If you have questions, I'm more than happy to put you in touch with them. We've gone gone way over, so thank you guys so much for your time and attention, but we'll wrap it up. Thank you.